Dear ladies and gentlemen, good afternoon, and I'm privileged to present to you a lecture on wide use of real-world medication data in Europe and its challenges. I apologize I cannot be with you live, and that's why I have recorded this presentation. My name is Miriam Sterkebom. I'm working with the Institute of Health Data for Unicom, and I'm also have an affiliation with the University Medical Center Utrecht as a professor in real-world evidence. So the outline of the presentation today is first to talk why do we need big data and collaboration to evaluate medicines. Subsequently, I will talk about two examples. First, the Safety of NSAIDs project, which ran between 2009 and 2014, and then an example from the IMI Advanced project that is about vaccines. Then we will talk a bit about the conception common data model, and then we'll draw conclusions. So there's clearly a large need to use big data to evaluate medicines. And I think 2004 was a pivotal year when Vioxx was actually voluntarily withdrawn from the market after 5 years of marketing, because it had been shown in a clinical trial that the use of rovacoxib could increase the risk for myocardial infarction. This led to the fact that this took 5 years and millions of people to be exposed prior to detecting it led to a review of the Institute of Medicine of the pharmacovigilance system. And they concluded that the way medicine safety was being evaluated should drastically change. In that review process, Richard Platt from Harvard Pilgrim Healthcare had calculated that if electronic healthcare data would have been available with at least 1 million subjects, that the association between rovacoxib and myocardial infarction could have been detected in 3 months. And this was the start of what is currently known as the Sentinel system, where indeed electronic healthcare data on 100 million people are actually being utilized to monitor safety for the FDA. And this is all funded through FDA and put in law by the FDA Amendment Act. In Canada, in 2013, the CNOTS project was set up and paid for by Health Canada, also to monitor medication safety, and it's actually a collaboration between the Canadian provinces that have very similar type of data, so it's actually quite good to pool them. In Europe, there was also a notion that collaboration should happen and that people should not conduct their own database anymore, but that you should actually be able to work together across projects or across databases. And the first attempt to do this was done in the EU area project, which was set up to be able to use electronic healthcare data to detect safety signals. And it used distributed analytics and a common data model. Subsequently, there were several formal medicines evaluation studies requested by the European Medicines Agency to look at class effects and, or the safety issues related to an entire class of drugs. Then afterwards, and that period, we also had the pandemic, the H1N1 pandemic, and subsequently many studies have been conducted, and the evaluation or the evolution of methods and tool has continued. So let's look at one of a good example. So the first example I would like to share with you is the example on the evaluation of traditional NSAIDs and coxibs and their risk of cardiovascular disease and upper GI bleeding. If you're interested in any of the publications around this with the data, so here you can actually find that. In the SOS study, 7 data sources participated, 3 from Italy, 2 from the Netherlands, Germany, and UK, together having access to a source population of about 35 million subjects, using various types of data from medical record databases such as IPSI and THIN, and PDNet to claims databases such as the CISAR and the OSIF and the BIPS database, and then a record linkage of different type of data in Pharmo. Now what you see on the right-hand side is actually the terminology by which the diagnoses are being coded. So you could see several use ICD-9, there is ICD-10 in Germany, READ in the UK, and ICPC in the Dutch general practice database. The model that was set up actually in the EUDR system that has been used for about 10 years across many different studies and about 40 different publications, I think is a quite simple common data model. On the top you see 3 files, and the local data access providers will need to put their data into this format. So in these 3 input files, a prescriptions file, an events file, and a population file. And then they can actually apply a program. And in this instance, it was Jerboa, but you can also run SAS or R. And the results of that program are actually being shared. And in this way, background rates could be calculated, incidence rates of events during use, or case control studies, and drug utilization studies. What you see here in the events table is actually an event type, which means that these files are both structurally, syntactically we call that, and semantically harmonized in some way. And the biggest issue in many of the initial studies was how to harmonize the event type, and especially if you saw the different terminologies that we were just looking at in the source data. Medicines were harmonized at ATC level. And so they were less of a problem because this was mostly available in most as a common terminology, many of the databases, except in the UK databases where they needed to map it to ATC from the BNF code. And then people would use their own procedure to calculate the duration of a prescription. So most of the initial hurdles were actually in how to deal with the heterogeneity in diagnosis recording, and especially in Europe, because in the US everybody uses ICD-9 and they use very similar type of data sources, both in the US and Canada. So especially in Europe, where we have all the different terminologies and languages, this was an issue. So within the EUDR project, there was an initial approach to harmonization of events, and this is the key publication you see listed here from Paul Avila, who is currently working at the Harvard Medical School. And it's based on the use of the Unified Medical Language System, which is a meta-thesaurus actually that allows linkage to the different terminologies. Now, within this paper, there is a stepwise process, like 6 steps that would bring you from the case definition to the final list of codes that you would need to actually identify events in a data source. Now, here you see an example of the complexity of what it can be from the SOS project that I used as an example. So in the SOS project, we had 4 outcomes. Upper GI bleeding, acute myocardial infarction, stroke, and heart failure. And there were 41 confounders that needed to be considered, and there was 1 exclusion criteria. So, and we had these terminologies across the data sources, and we needed to find a common semantic base. So that meant that these events were translated to UMLS concepts, and that then could be mapped out to the different codes in the different terminologies. And here you see for those, for the terminologies, what the different level in granularity is. So for READ, it's the most granular terminology, whereas, for example, ICPC is much less granular. And that is what is actually causing the problems. Now, one of the key steps in doing this is also the evaluation of whether this makes sense and whether you have completeness. And that was a very tedious job and often took very long. So that is why in the IMI Advanced project, which we talk about a bit more later, this process has been optimized or semi-automated, And now there is like a tool which is publicly available. You can find the link here where you can plug in a case definition. It uses text mining to search the concepts in the UMLS and then maps it out to the coding system. So this has been optimized. Here you look at the results, some of the results of this SOS study. So basically what you see here is for different NSAIDs, traditional and COXIPs, you see their in the odds ratio. So it means that everything above 1 actually means an increased risk, and that is for current use of that NSAID against past use. And you can see here a nice ranking with the highest risk for ketorolac and indomethacin, etoricoxib, rovacoxib. So very nicely showing that across, pooling across those 7 data sources, first of all, leads to the results we would expect. So we can confirm that rovacoxib, that dicoxibs have an impact on myocardial infarction. What you can also see is that there was actually an opportunity and an ability across all those data sources to look at a large range of NSAIDs, which is often not done when you look at a single database where you may only be able to have, estimate the most frequently used NSAIDs. But this allowed us to actually have many estimates. Here is a similar table for heart failure, and again here you see the high risk for ketorolac, etoricoxib, rovacoxib, so very much confirming what we knew, but also having the ability to provide estimates for a lot of other NSAIDs that otherwise would not have been able to be investigated. What SOS also showed is that we would have wished to actually look at dose and duration effects or strength, and this was only possible in 3 databases because of the limited type of information that was actually available in the others. So clearly improvements that are possible would actually be to have a better ability to look at other details about a drug beyond the active ingredient only. Example 2 is actually about the IMI Advanced project, which aimed to build an ecosystem to monitor vaccines following the H1N1 pandemic. Lessons learned, which showed it's really needed to collaborate in Europe to monitor vaccines. And of course, why do I take this example? Because it's very topical. And this will also happen for COVID-19 monitoring, that we need to actually have an ecosystem. So many of the lessons learned from IMI are now going into the building of the ecosystem to monitor COVID-19. The way we worked in this project is actually the distributed analytics. All data access providers, they have their data local. They convert their local data into the common input files. Here, a patient file, a vaccination file, and a diagnosis file, very similar to what you saw for the SOS study, but now it's vaccination. And now you see here that there was actually a variable which called VAC type, vaccine type, because many data sources were actually not able to even find an ATC code or a brand. We'll look into that. They had this, then they got a ARP program or a SAS program. They ran that, and the aggregated data were then shared on a remote research environment where people could connect to do the last analysis and pooling. So if you look at coding systems for vaccines, it's difficult. Here you see some where you could look at, but often you do not have enough detail to have all of the ingredients of the vaccine. So often we do know the type of pathogen or disorder it's actually targeting. Not in all strategies we know the vaccine strategy or the platform that is being utilized. So how is the antigen actually being presented to the body? Often in the ingredients are not always known, such as adjuvants, for example, in the ATC. And valencies are not always known in each of the terminologies. Now, this lack of detail on products will be really difficult for the COVID-19 monitoring. So I would really hope that we have IDMP and it will be recorded in some way. And here you see an example of like one of the vaccines that was being type of vaccines that were being looked at in the HPV in different data sources. So here you see the countries, the type of the data sources that were being utilized, the birth years included, the total persons, and then the information on brands. So Cervarix and Gardasil are the two key brands. And you see in many data sources, there is no brand known, actually. So only in very few you can actually know what type of vaccine is being utilized. So the way that we code medicines in common data models has evolved over time. And I would like to share with you the approach we're currently taking in the IMI-funded conception project, which is about medicine safety evaluation during pregnancy and lactation. And within this project, we have been building a new common data model, which is has many more tables than the 3 that we were using initially. And the concept has also slightly changed, meaning that we distinguish 2 steps: a syntactic harmonization, so where local data access providers transform the data into a structure that has same variable names and table names, and then the semantic harmonization. And now we actually focus on doing the semantic harmonization more centralized, as it was difficult to see what was locally done if that is immediately done by the data access providers. So now within this common data model, you see, you have a persons table with observation periods, person relationships, mother, father, child, ability to have primary data collection linked, survey observations, medical observations, visit occurrence, events, procedures, and then most importantly here, a vaccines table, a medicines table, and a products table that can actually have details about the different products. And then there is metadata, the instance, and the source of the common data model. I'm just going to look further with you at the medicine and products table that is currently defined, and we're still working to finalize this. But so this is the medicines table. You would have the person ID, the date of the dispensing, prescription, the product code, the local product code, the ATC code, and then this would be able to be linked to a products table where you have more detail on the product code, like almost like the pharmaceutical product actually. And so the key question that we are having now is whether IDMP would actually make this easier, or whether we could use these tables to immediately map to the IDMP, so that we can collaborate better and take the hurdle on the improved medicines harmonization across databases in Europe. So the conclusion for today is that medicines evaluation needs big data and access to multiple data sources across countries. We have learned that, and it's currently state of the art actually to do that. We also know that within Europe, data sources are very heterogeneous. There has been a lot of focus to overcome that heterogeneity and to deal with it through harmonization of event coding, but much less work has been done in medicines because it initially was possible to use ATC, but we are seeing that using only that is difficult in the vaccines, and often it also doesn't provide enough detail, and not the amount of detail that we would want to have. So I think that the next steps really, and that's why I think Unicom is very exciting, is that we need to take next steps towards harmonization of medicines at more specific levels for these type of studies. Thank you very much for your attention.